Okay, yesterday we were talking about DNA and RNA. There's actually three types of RNA. Messenger RNA, which we abbreviate as mRNA if you remember, takes the code from DNA and goes to the ribosomes to help make the protein. So it serves as a messenger from DNA to the rest of the cell. It carries the code and it can then read the code. If you remember from the other day, I mentioned how DNA cannot read the code. It has to give the code to mRNA. Then there's transfer RNA, or tRNA. It brings in or transfers in the proper amino acids to the ribosomes. It knows which amino acids to bring in because of the code for messenger RNA. You hook enough amino acids together, you get that protein that your cell is trying to make. There's also a ribosomal RNA, or rRNA. It goes along with the nucleolus to make the ribosomes. Messenger RNA and tRNA need a place to make the protein, and that place is the ribosomes, which are made by rRNA. So like I mentioned before, you have to hook enough amino acids together to make the protein. So amino acid plus amino acid plus amino acid plus amino acid plus amino acid, about 100 more, and you'd have the protein. Proteins get things done in your body, like making enzymes to digest food. As we learned yesterday, your DNA is in your nucleus. It will give the code to messenger RNA. That is the process known as transcription. You should already know how to transcribe DNA into messenger RNA. That takes place in the nucleus. Once the mRNA has been made, it is going to head to the ribosomes, because it is the ribosomes where protein is made. Once it's in the ribosome, it will start reading the code. tRNA will go get the proper amino acid and bring it to the ribosome, and the protein's made. Translation is a process that takes place in the ribosomes. It is the reading of the messenger RNA and tRNA bringing in the proper amino acids to make the protein. So rRNA, each of them plays a distinct role in the production of proteins. There are 20 different amino acids. You put them together in the proper order. That order would be the code that originally came from DNA, which then the code is given to messenger RNA. The instructions coded in DNA specify the order in which the 20 different amino acids are put together. So coded instructions, the genetic code. The code is written in a language of 4 letters. In DNA, it's A, T, C, and G. In RNA, it's A, U, C, and G. So this code, or in other words your genes, have 4 letters. The nucleotides in messenger RNA are read in groups of 3. So there's 4 possible letters, but they're read in groups of 3. So for example, messenger RNA could be read as UUA, then CGU. Each of those groups of 3 is going to form an amino acid. So right there, UUA, CGU would form 2 amino acids. Messenger RNA would keep being read in groups of 3 until it hits a stop codon and the gene is formed, or in other words, the protein is formed. A fancy term for a word in DNA and RNA's language is codon. Codon is a group of 3 nucleotides in mRNA that specify an amino acid. For example, UUA is a codon. CGU is a codon. A sequence of nucleotides in mRNA, it could be like AAA, CAC, CGU. Right there would be 3 codons. 3 amino acids were just made. You hook enough amino acids together, you get that protein, which is going to allow your body to function properly. This is something known as a translation wheel. You'll be using this to translate those codons into the proper amino acids so that a protein can be made. For example, on the last slide, we had AAA. If you look at your translation wheel, and if you look at A's, you start in the middle with the big A, so you have A, that group of three, or that codon, is going to make lysine. LYS is the amino acid that is made. So we will practice with this translation wheel in class. Translation is the process of taking those codes, the codons, and actually making the amino acid so that the protein can be made by your body. So each nucleotide in the messenger RNA are decoded into a sequence of amino acids. With translation, it's the language of codons into the language of amino acids. Each codon represents a different amino acid. And remember, a codon is a group of 3 letters in messenger RNA. So with messenger RNA, with AAA, there's one codon. CAC, there's a second codon. GGU, there's the third codon. We're going to make 3 amino acids. If we're doing the process of translation, it's going to be translated into lysine, which I showed you in the previous slide. CAC is going to form histidine. GGU forms glycine. And it's the transfer RNA, or the tRNA, that's bringing in the lysine, the histidine, and the glycine. Ribosomes read the codons in messenger RNA in groups of 3. Remember, messenger RNA was made in transcription. It then allows the transfer RNA to bring in the proper amino acids so that we can make a protein. So a section of DNA directs the synthesis of a protein. So the end game in all this is to make a protein. Hundreds of thousands of proteins are made in your body all the time. It's keeping you alive and helping you to grow. Genes within your DNA are codes. The order of the letters are the code, and it's a code for protein. Many proteins act as enzymes or helpers. Here's an example if we're talking plants. A gene could control flower color by coding for a particular enzyme that makes red pigment. The proteins from other genes regulate the growth and function of the organism. By coding for proteins, DNA holds the key to life.